Okay, so good morning. Yes. Okay, so I'm going to talk to you about the organization of transcription in nuclear space. And of course, yesterday we have already heard from, for example, Simon, that nuclear space is highly organized. And I want to show you now that this is also true for components of the transcriptional machinery. So here you see a still of a movie made in Ibrahim Sis's lab where they beautifully show that RNA polymerase II is not uniformly distributed through the nucleus, but forms these clusters, big and small. And when we look at transcription factors, in this case UBX, an important transcription factor in Drosophila embryos, we can also see that it's highly clustered. I want to note that in this case, they had to use expansion microscopy to actually see these clusters, because very often they're tiny. And so many people have seen in many systems that RNA POL2 forms clusters, transcription factors form clusters, but it's not so clear how these accumulations of machinery form, if genes come together in these clusters to be regulated, and related whether these transcription bodies actually have any impact on transcriptional activity. And I think one of the reasons that this has been really difficult to study is because, as I show you, they're often very small. There's many. It's not so, they appear in unpredictable manners, and often we don't know what the underlying sequence is of these transcription bodies. And so this makes it really hard to study them functionally. And so here comes the zebrafish embryo. And before I tell you how we use the fish embryo to address these questions, I want to give you a quick primer on fish development. So here you see the first couple of stages of fish embryogenesis. And what's important for the talk today is that upon fertilization, there's no transcription in the fish embryo. And this is true in most animal species, and actually also in plants. And development during these stages is driven by maternally loaded RNA and protein, here indicated in red. This over time gets degraded, both actively and passively. And at some point, transcription then begins, and the embryo takes control of its own fate by using its own genome. And we've been using this transition from maternal to zygotic control to address a series of questions in transcription regulation. And while looking at this, we saw a couple of years ago that when transcription begins in the fish embryo, it begins in two areas in the nucleus. And so this was sort of surprising. You can see here the elongating form of RNA polymerase II, so POL2 SER2 FOS. We look at this with fragment antibodies, which we can use to live image transcriptional activity. And you can first see these two spots, and then over time, as you can see, transcriptional activity spreads through the nucleus. And so these transcription bodies are large, they're isolated, and they appear in a very predictable manner, which solves some of these problems that I highlighted before. And so here, I'm showing you this because I thought it's a nice view at the embryo, and you can see these transcription bodies, but it's not very clearly visible. Perhaps on the TVs it is. But so these two spots, bless you, appear very reproducibly in all nuclei at the same time in the embryo. And so we now use these to address some of these questions that I raised. And this is a story that was already published, so I just want to summarize it, but I think it's interesting to get a sense of what you can do with these transcription bodies. So here we took these two transcription bodies that have elongating POL2, that you can see, actually I have a pointer, that you can see here. And then we asked, when we look back in time, how do these transcription bodies actually form? So what appears on site first, and then how do these recruit machinery? And we found that in the case of these particular transcription bodies, we first see NANOG, an important transcription factor, cluster at these sites. Those then, NANOG then recruits SOX19B, another important transcription factor, and this, they are both required for the recruitment of RNA polymerase II and the onset of transcription. And we were also able to show that of this NANOG protein, you need both the DNA binding domain and at least one of its IDR, so intrinsically disordered regions, to recruit additional machinery to these transcription bodies and activate transcription. And I'm showing you this because I think it's, it's helping us to start and get an idea how transcription bodies actually form in the nucleus. One more thing about that, we now know that NANOG seeds the formation of these transcription bodies, but when we looked even earlier in time and asked how these bodies actually form, we saw something very remarkable, which I hope you can also see here on the screen. So here, this is a nucleus. We're labeling nanog in green here and transcriptional activity in purple, and you can see many nanoclusters. And to prepare you for what's coming, the nanoclusters that are indicated with, are highlighted with these arrowheads are going to move together, and only when they're together does transcription start. So I'll show you here. So they're moving, moving, moving. And then you can see purple, which means that the transcription of this locus starts. And we're really fascinated by this movie because I think what we're seeing here is clusters of nanog that form on pieces of DNA that come together to activate transcription. And we have evidence to believe that transcription really only begins when a minimal amount of nanog has accumulated in this one spot. And so I hope next time perhaps I can tell you more about this process. Today I want to talk about perhaps the most important question surrounding these transcription bodies, which is, do they actually impact transcription? And if so, how? This is work spearheaded by Martino Ugolini, who's also here. He has a poster this afternoon, and so you can talk with him later if you want. And he worked on this together with another graduate student, Ksenia Kuznetsova. And just for those of you that don't think about transcription bodies every day, the current thinking in the field is that there's a locus, here in blue, for example, that recruits a lot of machinery, transcription factors, RNA polymerase II, potentially also other genes, and then those other genes might benefit from this local high concentration of machinery to rely on that for their transcription. And so this is the most accepted model, but there's not all that much evidence actually for this. And so we wanted to use our transcription bodies to address the question of functionality of transcription bodies. So again, you've now seen this a couple of times. We have these two transcription bodies that form in a very isolated manner, and we know, I didn't tell you that yet, what sequence seeds the formation of these transcription bodies. And so we can now remove that sequence, and then in this deletion mutant, we don't see these transcription bodies anymore. And so now we can simply take embryos that have these transcription bodies, embryos that don't have them, and ask, is gene expression affected beyond the expression of this specific gene? So are other genes impacted by the loss of these transcription bodies? Before we do that though, I need to tell you about one trick that we had to play, because of course by removing the locus that seeds these transcription bodies, we do not only remove these two transcription bodies in the nucleus, we also lose the mature microRNA miR430, which is the microRNA that is encoded in the locus that seeds these transcription bodies. And so this microRNA is super important for fish development. In fact, it helps in the degradation of maternally loaded RNAs, and so we need to bring back this microRNA somehow to not impact embryonic development beyond the loss of these two transcription bodies. And so the way we do this is we inject in the fish embryo the mature microRNA, so that we can now investigate just the loss of these transcription bodies and not the loss of these microRNAs. And just to show you that this works, if we now inject an RNA, so RNA encoding for GFP with 3 target sites for this microRNA, you can see that in the mutant, we see expression of this RNA because the microRNA does not cause the degradation of the GFP RNA. But when we inject these embryos with the mature microRNA miR430, we lose expression again. And so this is to suggest that we do get back the function of the microRNA. And further evidence for this is that the microRNA mutant has important developmental defects. We'll not go into the details of what exactly is happening, but when we then inject this microRNA, development looks exactly like it does in wild type and heterozygous fish. And so in fact, these fish that have lost the two transcription bodies develop into very normal looking and fertile fish. And while this is cool, because now we can use these fish to ask what the role is of transcription bodies, it was also kind of disappointing because of course we had hoped that these transcription bodies and their loss would have impacted development somehow. So I'll get back to that in a second. Okay, so what is the effect on transcriptional activity, if any? And so just to highlight this again, we're comparing wild-type embryos with these mutant embryos that we have supplemented with this microRNA. And the method that we used to analyze gene expression changes was what we like to call enriched-seq. And this is important. We cannot just do RNA-seq, because in these early fish embryos, there's a lot of maternally loaded RNA. And so when we sequence them, we have a really hard time detecting the very low amounts of zygotic transcripts. And so we inject 4-thio-UTP, which we use both to enrich for zygotic transcripts and also to see them in sequencing by converting them. And so using this technique, we found that actually there's a lot of genes that are misregulated. So there's a lot of genes that are downregulated in the mutant, and there's even more genes that are upregulated. And so that's cool, because that suggests that these transcription bodies have an important role in gene expression regulation beyond the locus on which they are seated. And so when we look at these genes or their distribution, you can see that they are distributed pretty uniformly across the genome, which was interesting because you might have expected a very local or in cis effect on the chromosome where this transcription body is forming. Actually, I think I didn't tell you that, but this transcription body forms on chromosome 4, where the miR430 locus is coded, so it's highlighted here. Okay. And before going into the potential mechanism of how transcription bodies impact transcriptional activity, I want to come back to this issue that we do see differences in gene expression, but not really any development of phenotype. And so to look at this, we asked these genes that are misregulated up or down at the developmental stage at which we did our initial experiment, how does their expression develop over time when we compare it to wild type expression levels? And as you can see, in 4 cell cycle stages, the expression of these genes returns to normal. This means that these genes, they are misregulated, but over time their expression sort of recovers very quickly. And just to say that this is in the time frame of 1.5 hours, so these cell cycle stages are super fast. And when we look at that differently, so now not just at the genes that are misregulated at this early stage, but just asking how many genes are misregulated at different stages, you can see that the numbers go down significantly over time. And so, yes, there's a gene expression effect when you remove these transcription bodies, but it's very short-lived. And we think that this may explain the lack of an obvious phenotype of these embryos. Okay, then to the mechanism. So how does the removal of 2 transcription bodies impact gene expression? And these downregulated genes, their identification, of course, is in line with this model that I highlighted before. So if there are genes that come together in a transcription body to benefit from a local beneficial environment, if you remove that transcription body, these genes would now be downregulated. And so we think that the identification of these genes support this model. By no means proved that model, but we were more fascinated by the upregulation of genes, and so that's what I want to focus on from now on. And we hypothesized that perhaps what's happening is that in these transcription bodies, there's so much of the machinery that other genes in other places in the nucleus cannot be transcribed because machinery is sequestered in these transcription bodies. And so we went on to investigate this hypothesis. And the first question that we asked is, what are actually these genes that are upregulated in the mutant? And so we took these 700 genes and asked, when in the wild type embryo are they transcribed? And we find that actually they are transcribed very quickly in the wild type embryo after the time in which they are transcribed in the mutant. And so you can see that 60% of these genes are transcribed within 30 minutes after the time they are transcribed in the mutant, and almost 80% is transcribed within 1.5 hours after their time of transcription in the mutant, suggesting that it's not some random upregulation of transcription, but actually these are genes that are already ready to go, but are not somehow fully expressed. And here you can see that the level of expression of this group of genes in the mutant at an early stage and in the wild type at a somewhat later stage correlates very well, further supporting this idea that it's really just an advance in transcription and not some real misregulation. Okay, so next we looked at what the transcription start site of these genes actually looks like in wild type embryos, because we get the sense that genes are transcribe too early, but what do they then look like in the wild-type embryo? And so when we do a toxic in wild-type embryos and look at these upregulated genes that are not expressed in the wild-type, they're highly accessible at their TSS, suggesting that a transcription factor has already bound and RNA polymerase II has already bound, but it can just not go into elongation. And here I'm showing you for comparison the top 700 most expressed genes at that stage, and you don't really see any difference in accessibility. And I want to highlight once more that this is remarkable because these upregulated genes, when you look at their expression level, are not expressed in the wild type, and these genes are very highly expressed. And so the TSS is open, suggesting that whatever is sequestered in these transcription bodies is something downstream of transcription factor binding and RNA pol2 loading. And this is in line with the fact that we fail to discover any transcription factor binding site in these upregulated genes that is enriched. And just to explain the thinking, of course, if in these transcription bodies a transcription factor were sequestered, you would think that upon its release, genes with a binding site for the transcription factor will be upregulated, and we don't find any enrichment for any transcription factor. So we're more and more thinking that what is actually happening is that the genes, that what is sequestered is a pause-release factor. And that then reminded us of an observation that we did a couple of years ago, which we were never really able to explain. So I now showed you many times that in the early embryo we have these two spots of elongating RNA polymerase II. But when we look at initiating RNA polymerase II, we actually see that there's additional spots. So these are the two miR430 transcription bodies, but we have additional initiating spots, and these never go into elongation in the wild-type embryo. And so we never really knew what to do with this, but now we thought perhaps what's happening is that a pulse-release factor is sequestered in these transcription bodies, and as long as these transcription bodies are there, these ectopic transcription bodies are not going to go into elongation. And so the prediction of that is, of course, that if you remove these transcription bodies, you would now release that factor, and more initiating bodies would go into elongation. And so this is what we see. I don't know if you can see it, but I'll tell you what you're supposed to see. So in wild-type embryos, we have these two miR430 transcription bodies where we see initiating and elongating RNA pol2. We don't see any additional elongating RNA pol2. In the mutant embryo, we don't see these large miR430 transcription bodies because we have deleted the locus. We do see these ectopic initiating bodies, and lo and behold, some of them are also going into elongation, as we would have predicted, and we see the same at a somewhat later stage. And perhaps this is better visible. Here we have quantified many of these images, asking how many ectopic transcription bodies just have initiating pol2, initiating and elongating, or just elongating RNA polymerase II. And you can see that the yellow and green fraction is increased in the mutant background. And the same again for this later stage in development. Okay, so then the final question, I guess, is what is this factor that is sequestered, and can we prove that it's really present in limiting amounts and sequestered by the miR430 bodies? And so we chose to look at CDK9, which is a likely candidate when you're thinking about pulse release, and we can see that it is high in these transcription bodies in wild-type embryos, which makes sense because there's transcription elongation going on. It's very low in the rest of the nucleus. We don't even see the outline of the nucleus here. And then the question is, if we now overexpress CDK9 in a wild-type background, so where the endogenous CDK9 is sequestered by the miR430 transcription bodies, supposedly, can we now move initiating bodies into elongation? And so the experiment that we did was we took wild-type embryos and injected CDK9 mRNA. And again, so this is a wild-type embryo. These are now 3 different stages. In the wild-type, you can see these 2 elongation clusters only at the miR430 locus. In the wild-type embryo injected with CDK9, we also have these 2 miR430 bodies, but we see ectopic bodies going into certofos as well. So yes, the overexpression of CDK9 pushes some of these initiating bodies into elongation. And that phenocopy is what we see in the mutant, miR430 mutant, where we of course don't have the miR430 transcription bodies, but we have these elongating ectopic transcription bodies. In fact, when we quantify this effect, so here you can see the 3 different stages comparing the ectopic transcription bodies that have elongation in wild type wild type injected with CDK9 and the mutant, you can see that the effect of injecting CDK9 is almost stronger than what we see in the mutant embryo. Okay, so we think that this provides some evidence for CDK9 being present in limiting amounts and it being sequestered in the early embryo. Okay, so I'll summarize all that. In case you didn't see any of the images, you can now take my word for it. We remove these two large transcription bodies and we find that this causes a genome-wide misregulation of transcription. The downregulation of hundreds of genes we think is in line with this previously proposed model where genes come together to benefit from a local high concentration of machinery, but more work will need to be done to really characterize this better. The upregulation of genes, we think, might represent the underappreciated role of transcription bodies in sequestering machinery, and this might be an important general role of transcription bodies in transcription regulation. And here, this is a visual where the miR430 locus creates these huge transcription bodies that brings in other genes to be transcribed, supposedly, but it also sequestered machinery, and therefore these ectopic transcription bodies don't go into elongation. And upon the removal of these transcription bodies, now we get redistribution of machinery and activate genes elsewhere as well. Okay, so these upregulated genes we find are poised and ready to go already in wild type. In absence of these two large transcription bodies, these ectopic bodies go into elongation, and the overexpression of CDK9 phenocopies the loss of transcription bodies, suggesting that CDK9 is an important factor in this process. And finally, I also want to come back to this sort of conundrum that we do see an effect on gene expression, but not really on development. And we think that this is due to the fact that expression levels recover very quickly over time. And I think this has two important implications. One, that transcription bodies, while they might impact transcription, they might not form in a deterministic way, because clearly, if a gene normally is transcribed in the miR430 transcription body, it won't be transcribed there when that transcription body is missing, but it will be transcribed somewhere else. And it also suggests that the timing of transcription, which is sort of messed up in these embryos that we generated, is not all that critical in contrast to general belief. And so with that, I would like to thank the people in my lab, mostly Martino, who I said is here, and so you should chat with him. And yes, oh yeah, I also want to highlight Hiroshi Kimura from Tokyo Tech, because we get all our fragment antibodies from him, which are really important for the live imaging that we do. Of course, our funding, and I look forward to any questions that you have.
Yes, thanks a lot, Nadine.
Thank you very much. Can you comment on very early in your talk where you showed sort of the coming together of the NANOG guys, and then there was that preceded the transcription. I guess what I'm wondering is, can you look at whether the proximity to heterochromatin, like, is there does it proceed going towards heterochromatin with the NANOG, or does it? What is the progression of events with regard to sort of relative positioning in the nucleus?
Well, yeah, actually, in these very early embryos, there is no heterochromatin.
Oh,
and so we don't. Okay. No, I didn't mention that. Yeah. So, but it's for us, it's an advantage because it makes the imaging much easier. Yeah. But heterochromatin domains only appear later. I can't say it, thinking about. I'm not sure what you're headed to, but the progression, so these transcription bodies, they also resolve again. So when transcription begins and RNA accumulates, actually this transcription body explodes and explodes is a bit too dramatic perhaps, but yeah, NANOG leaves the site and the transcription body is gone. So it's interesting because RNA probably is required to form these accumulations, but too much RNA, as we also know from other work in phase separation, of course, causes the transcription body to dissolve, but I'm not sure that's what you were after.
We can talk. I have a reason to ask, but we can talk later. But your point about the early stage totally makes sense.
Thanks. Yeah, thank you. Great talk. 2 questions. So first, it's a bit surprising that you don't see any phenotype for the developing embryo. I guess it shows that it is a very robust system. So I was wondering if you have tried to challenge a little bit the embryo. Yeah. I don't know what can be done with fish, maybe changing the temperature or something like this.
We haven't, but of course this is something we're also disappointed, right? We would have liked to see some phenotype, but we didn't do anything of that sort, and so we really have to go into asking, when do you get an effect of this misregulation? Because there's clearly, I mean, we were pretty stringent selecting these genes, and so they really are highly up and down regulated, and so there should be some phenotype, I think, yeah. But we haven't done anything of that. It's a good suggestion.
And my second question is, do you think that this limitation of CDK9 is also at play in other cell types? Because
we don't know. That's also a really good question. I don't know. I actually think, I of course like to think that this sequestration by transcription bodies is a general principle. Actually, in Drosophila embryos, it has been shown that if you don't have ZELDA, RNA polymerase II actually moves to other places than where it would normally be with ZELDA. And so I think that there are other examples of where sequestration of machinery in the nucleus is very important in relationship to transcription bodies. I also think that CDK9 is not going to be the only factor that's sequestered there. We were sort of forced to test this, and so we focused on the easiest one, and it worked really nicely, but I'm sure that there's other factors too. And so we want to look in other systems, and also we want to be more comprehensive in understanding what is actually in these transcription bodies. At the moment, we only look at we look at the things that we think can be there, and so it's a very targeted approach. And now we're trying to do proximity by attenuation to ask what is all there, etc. So there's some work to be done. Yeah.
Thank you.
Oh, well, first, really beautiful work, and just to find any signals at all of spatial character on transcription, I think, is very important. But one thing that I'm sort of confused by is that, this system with NANOG is a gene that's part of a fairly complicated network. That network's been investigated on its own and has multiple steady states, let alone pathways between those steady states. And I don't understand whether you can really sort of disconnect those other genes from this in your analysis. You have ones that are connected, upregulated, downregulated, but actually there's cycles of regulation where something gets upregulated, which in turn downregulates something else. And, I don't quite see whether that's been settled. I mean, obviously there's much work to do. You could study multiple genes and see whether they're in the same place and so on.
I think I understand your concern, but I think that one benefit of doing this in the embryo is that we have a pretty clear understanding of which genes turn on when normally, and also the steps in time that we take are super short. It's one cell cycle after the other. And so I don't really think that we're looking at secondary effects, because that's, I think, what you're concerned about, right? Yeah. So I think we have a pretty good handle on the temporal control here, because we look at every cell cycle so I'm not concerned about that. It's the very beginning of transcription in the embryo, and so it's the first genes that are transcribed that we see being misregulated. So there is no secondary effect.
I just would, thinking about trying to model it, it would seem like there's lots of unknowns, like what actually are the rates when the concentrations have been upped by being in the body and so on. So I just don't really know whether
I totally agree with.
That sort of bulk measurement would be enough to tell you.
Yeah. No, it would of course be very interesting to know, first of all, what, maybe that's what you're alluding to, what's actually the fraction of protein that is in these clusters, how does it deplete the protein from the rest of the nucleus, how does this impact transcriptional activity? I agree, but it's the beginning of, I guess, that work, yeah. Thank you.
Hi, nice talk. I'm wondering, in your experimental setup, you took it out, the MIR430 locus, and looked at what happens. I don't know whether it's possible. What happens if you introduce an extra?
Yeah, we were trying this. We first tried to make deletions. It was very difficult because this locus is quite repetitive. That's why it's sucking up a lot of machinery initially. And so now we have taken small parts of that locus and put it elsewhere in the genome. But we've made these lines, but we haven't really analyzed them yet. But it's a really interesting question, of course. Or even looking in the heterozygotes, right? Because you would expect already differences there too. Also to get to this concentration question, I think. Yeah, thanks.
Thanks a lot.
Thank you.